Welcome back to MicroVerse. In this previous lecture, we distinguished between pathogenicity and virulence and introduced the concept of a virulence factor. We learned that microorganism cannot produce disease simply by being present in the host. It first overcomes several barriers and establishes successful interaction with the host. And in many infections, the first important step is surprisingly simple. The microorganism must attach. So today's question is, how do microorganisms attach to the host cell? Let's understand the process of microbial adhesion. So what is adhesion? Before microorganism can invade tissue, multiply, or produce toxins, it often needs to remain at the appropriate site in the host. This is where adhesions comes in. Microbial adhesions is the process by which microorganism attach to the host cell or surface. This interaction is not random. Microorganisms possess specific structure or molecules called adhesins, while host cell contain complementary molecules known as receptors. So, at the simplest level, microbial adhesions plus host receptor gives attachment. So, why is attachment important? Imagine bacteria entering in the urinary tract. If they cannot attach effectively to the epithelial surface, they may simply be removed by the continuous flow of urine. Similarly, microorganisms entering the respiratory tract encounter mucociliary clearance, which can physically remove them from the airway. Therefore, successful attachment helps microorganisms remain at the site of infection despite the host's physical defenses. Once attached, they have a better opportunity to multiply and establish infections. So, how does the attachment occur? The interaction between an adhesion and its receptor is often highly specific. You can think of it as a lock and key type of interaction. The microbial surface carriers and adhesions. The host cell present a suitable receptor. When the two recognize each other, the microorganism can attach to the cell. This specificity also helps explain why some microorganisms show preference for particular tissues. For example, microorganisms may colonize one epithelial surface efficiently but have much less ability to attach to another. This contributes to tissue tropism. So here we see the structure involved in adhesions. Different microorganisms use different structure for attachment. In many bacteria, fibri or pili can carry adhesions that help them attach to the host cell. Some bacteria also use surface proteins as a adhesions. In addition, certain microorganisms produce extracellular polysaccharides that contributes to attachment and biofilm formation. So when you see the term adhesions, don't think of one particular structure. It is a functional term referring to microbial molecule that participates in attachment to the host. So let's take an uropathogenic E. coli as an example. This bacteria possess specific adhesions that allow them to attach to epithelial cells of the urinary tract. This attachment helps them resist being washed away during urination. Once they remain attached, they can multiply and establish infection. This is one reason why adhesions is considered an important virulence mechanism. So there is another important consequence of attachment. When microorganisms attach to a surface and subsequently produce an extracellular matrix, they may form a biofilm. Biofilm are particularly important in clinical microbiology because they can develop on surface such as urinary catheters, prosthetic devices, and other medical materials. Within a biofilm, microorganisms may become more difficult to eliminate and may show increased tolerance to antimicrobial agents and host defenses. So, a process that begins with a simple attachment can eventually contribute to a persistent infection. So, now let's connect today's concept with the previous lecture. We define a virulence factor as a microbial property that contributes to the ability to cause disease. Then, adhesions are therefore important virulence factors. Without successful attachment, many microorganisms have difficulty establishing colonization or infection. So our sequence is becoming clearer. Adhesions, then colonization, then infection, at last disease. Of course, not every infection follow this sequence in exactly the same way, but adhesion is an important early event for many pathogens. So let's quickly bring the concept together. First, adhesions. Adhesions means microbial molecule involved in attachment. Then second one, receptor. Receptor is the host cell molecule recognized by the adhesions. Then adhesions means attachment of the microorganism to the host. And the key idea is no effective attachment means difficult to colonization. So today's question was, how do microorganisms attach to the host cell? They use specific microbial adhesions that interacts with complementary receptor on host cell. This attachment allows microorganisms to resist physical removal, establish colonization, and in many cases initiate infection. But attachment alone does not necessarily produce disease. Once a microorganism has successfully attached to the host, the next challenge is, can it enter the tissue or survive host defense and spread? This brings us the next step in a microbial pathogenesis, which is invasion and tissue penetration. So, in our next lecture, we will understand how microorganisms invade the host and overcome its protective barriers. So, thank you for learning with MicroVerse. Keep learning, keep growing, and let's master microbiology one concept at a time.